Okay, so we're going to go ahead and get started. So welcome everyone. So thank you so much for joining us today. My name is Crystal Ramos Iniguez, and I am one of the program managers here in the STEM department, and I'll be your host for today's event. This presentation is the first in our series called First Generation Latinx Journeys in STEM. These guest speaker events were created to offer the opportunity for you to explore and learn about different pathways within STEM and hear from current professionals. Before I introduce our wonderful guest speaker, I want to go over the format for today's Zoom talk. The talk will run about 20 to 30 minutes, and then we will have a Q&A, a question and answers for the remainder of the time. If you do have a question during the talk, I would ask you to please type it in the Q&A box and do not unmute your microphone. Your questions will be asked by me in order received after the speaker is done talking. Nicole will be moderating the chat, and we'll make sure that everyone's microphone is off during the talk. If you have any other question that is geared to the actual format of today's event, please message me directly, okay? And now it is my pleasure to introduce our first STEM guest speaker in our STEM guest speaker series, Stefanie Brizuela. She is a first-generation queer Latina and second-year Ph.D. candidate in the Molecular Biology Interdepartmental Doctoral Program at UCLA. And she will be sharing her journey with us today. So without further ado, please give a warm welcome to Stefanie Brizuela.
Thank you, Crystal, for the nice introduction, and thank you to everyone who is attending today. So what I want to say about this presentation is that it took a lot of introspection. I think particularly in that sometimes scientific presentations can be really hypothesis driven, very techniques, a lot of jargon. And what I really wanted to share was how did I navigate a space where my parents are immigrants and I was the first one born in the United States. And so I really want to start off with the journeys that came before me. So my abuelita immigrated to the US in 1985 because of the Salvadorian Civil War. And when she did that, she actually left my mom and my tío and my tía behind because she wasn't quite sure how to navigate the risks around her, and she didn't want to impose those risks on her children. So later on, she brought my mom to the United States. And my mom had me at 17, so it was very hard for her to provide stability. And that's where my abuelita came in and just kind of provided the stability I needed, the care I needed, the attention I needed. And we actually had an arrangement where when my mom wasn't able to take me to school, she did. But it was contingent on I would have to teach her the lessons that she couldn't afford to do as a child. So what I mean by that is my grandma never got to finish elementary school. So when I was growing up, anytime I was in the car, like if I thought about how were mountains forming or why do fish breathe underwater, there were things that my grandma wanted to know about the world that really weren't taught to her more so than the observations. But at the same time, I realized that she was actually my first scientific role model. And so oftentimes I think when we grow up and we see things like, Albert Einstein on TV or Bill Nye, these are people that we think are scientific role models. But if we really boil it down to what it means to have scientific inquiry, it means engaging with the world and making observations, adapting to circumstances, trying new things and failing at them. And so for me, that was really inspirational that, she was a woman that I looked up to that took on the risks. They were, things that were very unknown to her, but she was able to create a better future for future generations like me. And so when I think about being a first generation student, I often think of I'm being the first in my family to be an American, as I was born in the United States, but I'm also a first generation high school graduate. And so what you guys don't know probably is that I'm a twin and twins run in the family. And the reason I'm pointing this out is because I think innately there was always that competition with twins. You're consistently compared. And not only that, but there was these cultural norms that my family was carrying from El Salvador versus the US soil where I was now growing up. And so there was a lot of confounding factors that were happening between trying to figure out who I am in my identity. I have a twin, but I'm also evolving as my own person. And so with that, I proudly do say that I'm queer. And the connections that I formed with people, I think have also become my family in a very non-traditional way because there have been a lot of rifts that have happened in my own family. But going into my journey of discovering myself and in STEM, in high school, my sister collapsed while we were doing homework. And this is when I started recognizing that there was not a little, actually, it was a substantial mistrust in the medical community. I mean, even growing up, if I was sick, I wouldn't go to the doctor unless it was something that was severely wrong. And I'm sure maybe some of you guys resonate with that. So when my sister collapsed, it took a lot of convincing to try to get my family to call the ambulance because I just saw that her lips were turning blue, like she couldn't really talk. So this was more than just a headache. And so we actually went through 3 different hospitals and even in helicopter where she ended up at the University of California, San Francisco. And it was there that they diagnosed her with arteriovenous malformation. Now, I don't expect you guys to know what that means because I didn't, and I kind of still don't. But what I can say is that there was a lot of questions that immediately come to mind, like, what is it? Why does she have it? Is it hereditary? Do I have the chances of getting it now? Is there a cure? Is there a treatment? And were there earlier signs or symptoms? I mean, if there's such a complex name for it, clearly there was research done. So what's going on here? But I think in that, it really taught me to become a scientific communicator. Because oftentimes we hear healthcare workers speaking in a language that is familiar with their colleagues, but it isn't necessarily familiar with our families. And so, in that way, I think it perpetuates this cycle of having a mistrust in the community and not exactly understanding what's going on. Especially, for example, if you pick up a prescription and they hand you a piece of paper and you read it, and you're like, okay, well, I don't really understand what's being said here. I mean, the risks, I don't know. But I think when I started kind of realizing, okay, I want to make sure that my family feels comfortable with what the healthcare workers are doing, I think I was 16 years old when I started reading scientific papers because to me, it made me feel disconnected, but I wanted to help my family in any way that I can, even if I was just a high school student. And so the way I think about arteriovenous malformation is that there is a gene mutation that causes a bypass of the blood that is going towards the veins and the arteries, and there's capillaries. In another sense, if we really apply this in a real world metaphor, when we have these highways and you have a slowdown ramp after the freeway, that slowdown ramp is there for the safety of other people and it's there for the safety of transportation, but also yourself. And so when we think about arteriovenous malformation, that slowdown ramp on the freeway is not there anymore. And there's a way that it's bypassing it. And so you're just having shunt force the whole time. How this pressure was building, I don't know, but I think it was really this moment that sparked my curiosity for trying to highlight these gaps that included not only a cultural gap, but linguistic, socioeconomic, and I think all these barriers that come with not only being the first generation high school graduate in my family, but even just understanding science innately for the first time and how it's applicable to the real world. So I guess my journey of discovering myself in STEM was that I proposed a major in bioengineering and a minor in bioinformatics. And when I applied to UC Santa Cruz, one thing I want to highlight is that it wasn't particularly for the prestige of the institutions. I think oftentimes when we're applying to schools, we think that, oh, they're ranked #1, or oh, they have the best program. Oh, I want to go to Harvard. But the reality is that you have to celebrate the wins that you have. And so for me, being able to graduate high school at all was a huge feat. And so going to Santa Cruz, I had never visited. I know that some students get to go to orientation, but I didn't have the stability in my family to be driven 6 hours away from Bakersfield to go view the campus. So I just got dropped off the first day and they're like, bye. And I'm like, okay. So now I have to figure out how to navigate the dorms and what financial aid looks like. And so it was a lot. And in addition to that, choosing my major, I figured that I liked math and that at this point I really liked reading science papers. So in my head, I was thinking I'll just combine them, and it'll be fine. Like I'll just do bioengineering and a minor in bioinformatics. But I really had no idea what I was getting myself into because when I started doing more research, and just on Google, I highlighted all the boxes in red where I'm like, okay, I did calculus in high school. I did some general chemistry. I did some cell and molecular biology, but then everything else after that is beyond my scope. And I think that recognizing when that intimidation factor sets in is pretty instrumental because you can have a fixed mindset or a growth mindset. And so there are the things that you don't know yet, or you could frame it in a way that's where are the things that I want to learn about. So this picture is actually an example. I think me and Crystal took this class where we had to, it was a bioelectronics course, and we had to create like EKGs, and there was a Lego where you drilled a hole through it. And then based on the light, it was giving you your heart rate. So it was a very abstract course. And even the major in and of itself was particularly interesting in that it encompassed not only math and biology, but programming. And I'll touch more on how that experience played out. But reading these science papers, communicating to my family, and knowing that I was going to start my undergrad for the first time, I really wanted to know what happened outside of the classroom because I saw that I was going back into a classroom, but reading science papers was something I was doing on my own time. So my journey of pursuing research started with me just Googling about opportunities. So I was already receiving financial aid, but I did take out quite a bit of loans. But I wanted to see if there was any opportunities out there where that I could support myself and be self-sufficient. And so I found this program called the National Institutes of Health Undergraduate Scholarship Program. And I looked at their requirements. I'm like, okay, 3.3 GPA, enrolled at a four-year university, demonstrates that they need financial aid, personal statement, research proposal. I figured like, okay, I got this. I didn't really look at the stats because I think oftentimes in the communities that we're in, and even just being in an underrepresented field or just marginalized educational infrastructures, I think oftentimes we count ourselves out of these opportunities, thinking that we don't have the level playing field of other people that got to go to private schools or other people that had tutors growing up. So the worst thing they can say is no, is the way I kind of like to frame things, is that it's not going to hurt me physically, emotionally, maybe a little bit, but I'll get over it. I think there are these moments where you have to accept failure in order to progress. So when I applied at my first year at UC Santa Cruz, I was rejected and I was totally fine with it because I knew that I was young, I didn't have experience. And I figured, okay, let me go talk to the MESA director at UC Santa Cruz. And I decided to take some time to prepare my next application. And the way I was going to do that was, okay, there are so many, this school is pretty big and I'm not quite sure what I'm doing. Does she have any advice for the things that I don't know about for this school? And so she directed me to her colleague at the Genomics Institute. And I think that was the first time that I was really understanding, like, okay, this is what science is. But when I signed the contract for this first summer internship, I didn't realize that it was a big data and data science internship. So, true story, I remember I signed the contract and then I really read what it was about. And then I was already at my dorm. And then I ran back to her office and I'm like, hey, actually, I don't know if I want to do this because I don't have programming experience. I have never done something like this before. This is a huge program. I don't want to let you down. And she just told me like, that's what research is though, right? Like you're consistently learning, you're consistently trying new things, and it's not going to be perfect, but you're going to learn so much about not only the research, but yourself. And so I decided, okay, I'm going to go ahead and go on Google and learn how to code for free. And I will say that a lot of my codes for the first time was like, if you guys have ever used Code Academy, it's it shows you, it tells you like, okay, you're at a movie theater and then you're going to buy these tickets, but you have to sort them like with Batman or Superman or Wonder Woman. And so when I was first coding for my science project, a lot of there was a disconnect between me and my mentor, because they would look at my code and say like, Stefanie, like, why does your code look like you're organizing lemons and strawberries into baskets? And I'm like, because that's kind of how I'm understanding how to organize my data right now, because that's how I'm learning on my own. But at the same time, it was this experience that made me want to think that things were tangible, right? And so this picture of me was the first time I was presenting research, and I carried a yo-yo with me. And it's very unorthodox. I don't think you'll ever really see that when you go to these national conferences or even school symposiums. But the reason I did it was that for me, I heard about beads on a string, and that's how DNA was organized, but I didn't necessarily agree with that metaphor. For me, it was more so like there was this organization around a yo-yo, and if I told you to guess how long is the string of a yo-yo, you wouldn't be able to tell me if it's all wound up. But if I unwinded it and showed it to you, then that is the way that proteins in our cells are recognizing the DNA and are able to say, oh, okay, now I can see it. Now I know where I'm supposed to bind. When it's wrapped up, there is inaccessibility. And so the principle of this demonstration was really just that we have heterochromatin and euchromatin, and it's the way that transcription factors, like the people viewing the string, are able to recognize the sites that they're supposed to bind. And so this was the poster that I generated for the Genomics Institute at the time of my internship. But what I really want you guys to take away from it is that there is a lot of images that are used. I think this is when I really not only started to feel like a scientist, but also recognize that I'm a visual learner. When I'm doing computational biology, even till this day, it is complex. I find it very difficult because it is a black and white screen of numbers and names and things that I don't really understand. But being able to generate images helps me. And so taking it back and looking at a broader scope, I realized at this point that, okay, I'm studying replication and transcription and translation, and this is coined the central dogma of biology. At this point, I realized, okay, I'm looking at transcription. Perhaps I want to maybe look at different parts of biology. And again, it was more so going with an open mind, because it's a humbling experience to know that you think you know things, but you really don't until you're touching other facets of research. And so when this experience happened, I decided to apply a second time for the scholarship, and describe at least my experience. And so not only the very technical science jargon that you probably saw on the poster if I were to zoom in, but more so what was that experience for me coming from a place of being a first generation high school graduate? How was I able to communicate my research? And I think that is what the National Institutes of Health recognized, is that not only was I on my way to becoming a scientist, but I was also on my way to becoming a communicator and a role model that other people can look up to, because the role models I had were scarcely lacking in the scientific world. And I think the headline for the Santa Cruz Sentinel was really meaningful, because being the first in my family to graduate high school, till this day, I still look back on that. And I still look back at this title, and I still look back at old pictures, because it's always reminding me that there is a full circle moment there, and you don't forget your roots. There are places that I will probably go and continue to go where my reputation has to be carried a certain way, or the way that I communicate my research or do research has to meet an expectation. But I don't forget the roots that it took me to get there. And I'm also allowing myself to be patient with that. So going to the National Institutes of Health for the first time, this scholarship was a pretty big deal. I didn't realize that there were few students chosen every year. When I applied, I didn't really look at the stats. I just kind of decided to, I'm going to read some science papers and I'm going to come up with the proposal and I'm going to show how passionate I am about scientific communication, because being a scientist isn't unidimensional. You have to really reflect your own personality and your upbringing, and that is what really shapes being an immaculate scientist, like someone who is able to connect to the world and their research. And so I decided, like, my first summer, I wanted to do something tangible. And what I mean by that is computational biology is very abstract, a lot of coding, things that I don't know, I guess instant gratification, because you can always control Z things. You can always copy and paste things. But I also wanted to see, where was my data coming from? I keep getting asked to download data, but who's making it? And so my first summer, I decided, okay, I want to go work at the National Institute of Diabetes and Kidney Disorders. And I was working with fruit flies, which was really cool. And I think at this time, CRISPR technology was really at its peak in terms of just being a really hot new technology. And so that was very awesome. And then I got to do a lot of wet lab stuff, which was really me in the lab with goggles and a lab coat for the first time. And at this point, I'm realizing, okay, so this time I'm doing transcription work, but I'm also doing wet lab. My first summer internship, I was doing transcription, but I did it in dry lab. And so I had a few questions, right? Like, you really start to reflect, like, I'm passionate about science, but am I passionate about this specific field? So was this something I saw myself doing long term? Are there more areas that I want to explore? Because you're consistently refining what your focus is. And so I started to get curious about, like, okay, then all the computational tools that I'm using, then why do we design them? And so I decided that my second summer was going to be the National Cancer Institute, and I got to develop a pipeline where we looked at splicing and we looked at how proteins were made. And for those who don't know what splicing is, and that transcriptional process, if I told Crystal and then I told Tommy to bake a cake from memory, um, the odds are they might be making either totally different cakes, or if I told them make a vanilla cake, they might be adding different ingredients to the cake. Maybe Crystal adds a cup of baking powder and Tommy's adding like a cup of vanilla extract. Who knows? But the point I'm trying to make is that the way that our cells are reading blueprints to make proteins are very similar to the way that cells are also or the way we're having recipes to make cakes, because back then we didn't have recipes in an actual cookbook. They were oral recipes, right? Like, you pass them on from your ancestors to learn how to cook. And so the way that alternative splicing is working is that you have many variations of the way proteins are made, and in a way that making a cake can be done in several ways. And so what I was realizing from this experience is, like, oh, okay, so now I'm getting more of an in-depth understanding on mechanisms. And so, like, what else can I learn? Because there was a point where I think I was struggling with, well, what don't I know? There's so much out there. At the NIH, there's 26 institutes. And so having to choose from one every year honestly became very stressful. And I went to mentors, like the one in the picture, who's actually closer to you on the screen. Her name is Dr. Carla Easter. And I remember just going, and I didn't really know her that well, but I would go into her office and say, hey, can I just vent to you really quickly about, all the pressure I'm feeling, or like how to choose an institute, or even my research. And she was really instrumental. But not only her, there are so many mentors that I'm not showing in the picture that it really took a village to kind of get to where I'm at. And so taking what I know, though, is I learned transcription in wet and dry lab. I learned translation in a dry lab sense and making a pipeline. And so now I wanted to finish my undergraduate degree and apply that to a health human context. And so for my senior thesis, I did lung adenocarcinoma and looked at alternative splicing. And this is actually a better representation of what I was trying to say earlier with alternative splicing, is forming a sentence and having all these punctuation marks. If you take out some of those punctuation marks, you're consistently forming sentences, and the sentences make sense, right? It's not like it's nonsense, that they all have different contexts. And I think that's really the beauty of what biology is that it's very context specific, and it's what makes humans unique. And so when I was finishing my undergraduate degree, I felt really accomplished. And at this point, I was really starting to feel like I was catching my stride. I was going to go ahead and apply to graduate school, and things were going to go well. And so my journey figuring out the next steps was that I applied to PhD programs. And the desire there was that I want to do both. There is just something about being able to do experimental work and do computational work that to me just clicks in my brain and gets me excited about the data that I'm generating. And then I get to sit down later and look at my own data. And then if I ever have problems, I can look back and see what I did experimentally. So there's just this consistent feedback loop of learning. And I wanted to do it in the scope of gene regulation. But at the same time, when I do research, I don't want it to come from an elitist space. So what I mean by that is oftentimes scientific research, scientific circles, even when COVID was happening, there was a lot of mistrust that was happening in the communities. And that is a prime example of what happens when information isn't disseminated to marginalized communities that don't have access to vaccinations, don't have access to healthcare. And so my desire with that is not only do I want to do science communication, but outreach. And it's moments like this where I'm participating in the Bakersfield College STEM program that I believe is a part of my journey currently. And I'm consistently going to continue to do outreach in that I hope to inspire others to pursue their bold ambitions. But in my journey figuring out my next steps, I applied to PhD programs, and I figured with the NIH scholarship, I had two options. I could choose to get accepted into a PhD program and then begin graduate school. But even if I don't, it's fine. I can just do my NIH postbac because that is what the scholarship program provides. But that's not actually what happened. And it was a very, very hard pill to swallow. I got rejected from all schools. And then the NIH told me that my postbac was canceled. And so I was really freaked out for a bit because this isn't what I was envisioning. But I will say it was an incredibly humbling moment because you think you have it all planned out. I always thought I had a safety net, but that wasn't the case, especially when this was canceled. I had no income for 5 months. So I really had to try to navigate not only my savings, but at the time I was living with an incredibly nice landlord who let me catch up on rent later on. But I knew that if I was in a different space, I probably would have been kicked out of the home I was living in. And so I'm really grateful for the connections that I've made with people along the way. An example of that is I ran into a former classmate who noticed I was still in Santa Cruz, and he said, "What are you doing?" And I said, "Well, I have no job. My postbac got canceled and I'm just kind of hanging out with my dog." And he said, "Well, can I refer you to my company?" And I said, "Yeah, actually, that would be amazing. What do you guys do?" And he told me they do regenerative medicine, which I was really interesting. I never pictured myself in industry. And I got to work with CAR T cell therapy and like traumatic brain injury and sickle cell, all while wearing this suit that you guys are seeing in the picture. That's not a picture of me, but that's literally what I wore. You have to get certified to wear something like this. Um, but something that was really novel about this experience is that oftentimes, I'm sure all of you have probably heard about academia versus industry, and where's the money at? Where's the opportunities at? Where does it fit with your lives? Um, Another thing I learned was initiating activities. And so industry for me was pretty regimented, but with purpose. A lot of things are regulated by the FDA. And so there isn't a lot of room for exploration or ingenuity. You really have to follow the code that they have there. But at the same time, I noticed that different departments do different things. Just like Crystal is doing a different role, Tommy's doing a different role in this program. And so that's oftentimes how industry works. Where do you guys meet in the middle? Where I believe that in industry, I initiated Science Corner, and it was really cool because what I did was I went into science journals, I pulled information about sickle cell or about CAR T cell therapy, and then related it back to the lab so that we could all understand like how is our company helping shape cures? And that was really amazing. But at the same time, I also was told that I could begin a remote National Human Genome Research Institute postbac. And I was thinking, wow, this is amazing because I have a job right now, but now I have a postbac. It took definitely a meeting and a lot of organization to tell my NIH director that, like, I'm currently working a job right now and I just started, and I don't think it would look good for me to quit. I think I need to continue to have this experience because I'm working on some things there. And after some meetings, we organized that it would be a remote postbac, and it turned out well. Because I think, in a sense, if you think about the desires I had, I got to do wet lab in a different approach, and then I got to do dry lab in a different opportunity. And although they weren't together and they weren't interweaved, at least I had these two experiences that I was doing. And so compartmentalizing ideas was also a skill that I learned from doing this very, very unique experience for about 3 years. And if you notice the title or maybe have caught a few keywords, I looked at DNA methylation in acute myeloid leukemia, and that was really cool because I had some splicing background, I had some computational work, and this whole post-bac was remote. And so, mind you, Maryland is 3 hours away. So there were times where I had to wake up at like 5 in the morning to have a meeting or even present my research. And so I'm getting up at 4 to make breakfast. And it was just a very odd time, but I loved every minute of it. I think it really taught me about how do we navigate unprecedented times in in COVID, for example, or just in general, where like life just kind of throws stuff at us. So it really shaped what I was going to do next, because I really started thinking about graduate school again, because I had the experience in industry, I had the post-bac at NHGRI, and it was about 3 years that I was out of school because, going back to when I applied in 2020, I was rejected, and then I just became a post-bac and Minaris cell and gene therapy specialist, and I was rejected again in spring 2022. Um, and I just continued with my postbac in Benares. And I think the mindset that I had was that astronauts apply like 10 times before they can go into space. They apply a lot. And I wasn't going to let anything kick me down, especially considering that with the with the merits that I have with the NIH and with my outreach experiences through Google or through the Strong Women, Strong Girls, or even being a mentor, I wanted to not only be better for myself, I wanted to challenge myself so that I can inspire others to do the same, so that you don't quit when things get hard. But mostly it's a point of introspection. And so that's what I mean by connecting the dots. So when I applied, I actually got accepted into UCLA, which was honestly, it was a dream school. I thought I was shooting pretty far with the school, but at the same time, I knew that my third time I was going to get in somewhere. And I actually did. I also got in at USC, and I also got in at other schools. So it was really amazing to tour those campuses for the first time. And if you notice my home area, it's gene regulation, epigenomics, and transcriptomics. And this really resonated with me because I was already looking at differential gene expression and processes and like splicing and computational work and like looking at different types of cancers. And so to me, this fit the scope of what I was doing. And it was really amazing to finally be accepted into this program. And I just want to go ahead and kind of show you a quick overview of what my scientific research looks like now, because if you've noticed in the past, I focused a lot on just transcriptional mechanisms, but what I'm really doing is that I'm in a signaling systems lab that focuses on innate immunity. And so what I mean by that is that we're looking at how innate immune responses, like our immune system, are responding to different stimuli. So if one of us gets sick and the other person gets sick, 3 months from now, we're exposed to the same virus. Maybe I'm responding better, but you're responding worse. And so it becomes really context specific. And I didn't have a background in immunology when I started in this lab, and this is actually the lab I'm staying in throughout my PhD. But what I really found interesting was that when we look at interferons, which are these like frontiers that regulate immune responses, we have the newest edition in 2003. And in 2013, we also have interferon lambda 4. And that was really interesting because, as we can see, things in biology are consistently coming up. And so being able to be a part of a group that is investigating this, where it dates back until 1957, is incredible. And so this overall, what I want you guys to take away from this is that everything in biology is complex. And this is an example of what the immune system is doing, where if we focus on these like kidney bean shaped yellow things where it says IRFs, those are transcription factors. They function as transcription factors. And what we can see is that when we talk about the interferons on the other side that I've mentioned, and then we look at the TLR4, that stands for toll-like receptors, they're responsible for viruses and different pathogens. Like, they recognize these patterns on these pathogens. But if you notice the middle, there's that pink squiggly line. Things are interconnected. And so I think that's what's really fascinating for our lab, was being able to draw those dots that connect those transcription factors. And more specifically, going to the TLR4 that I mentioned, when it's stimulated, I just want you to draw your eye down to the IRFs down below where it says IRF3, 1, and 7, because what we are finding is that when we have human macrophages, which is a type of immune cell, and you expose it to interferon gamma, which is a type 2 interferon, and then you antagonize TLR4, just like we saw these two pathways, we have interferon 2 and TLR4. Now, when you have them combined, they are affecting expression of this type 3 interferon, which is particularly fascinating. This isn't something that people have done before. And there aren't a lot of papers on it. And I've referenced the paper that it comes from my lab. And so some data I'm going to be showing you is something that I've done in the lab, where I just wanted to see if I can replicate the findings. And we found something pretty novel, where that even if you don't stimulate macrophages with interferon gamma priming, you can still see type 3 induction with interferon lambda 1. That wasn't seen here, right? Like, if we look at the gray naive lipid A line, we can see that it's right at the 0 mark, but that's not what I was seeing here. Although they are different techniques, this is something that I'm actively investigating right now, as that it's not exactly replicating the findings, but it definitely opens another avenue into transcriptional networks. And so what I really wanted to say at the end of this is that my journeys shape my PhD experience. I really do want to be a future and current role model that inspires individuals from marginalized communities to embark on this journey of self-discovery, because ultimately being a scientist isn't just about who you are in the classroom. It's really about who are you outside of the classroom, who are you outside of the lab, and what perspective are you bringing to the table, where you're making connections with new people in the lab, or the way that you speak about your research that makes you so passionate. And so for me, this is actually a visual of something that I'm submitting for a fellowship, where I'm working on these receptors. And like I said, I really go back and realize I'm a visual learner. And that's okay, because I think when you use different modes of learning, like kinesthetic or visual or auditory, it transcends cultural barriers, just like the way art does, right? When we look at art, you don't necessarily have to know the language if someone doesn't speak your language who's the artist. You just can look at it and really soak it in. And so for me, being able to do a graphical abstract and be in a lab that encourages that has been really instrumental. But it's also recognizing that I want to continue doing outreach. And so right now I'm working with you guys, the Bakersfield College STEM program, but I'm also signed up with the Kern County Women in STEM, where I've organized events for November and December to drive out to Bakersfield and participate in biology activities alongside people in classrooms. And then there's another program called Skype a Scientist, which is an international scale. So you can sign up and then they match you to a school. And so right now I am working with a teacher in Wisconsin who has newcomer immigrants, and they speak altogether 15 languages, which I think is incredible, but they also have disabled students. And I think for me, being able to go to a class of, I think they're in third grade, and try to spark their curiosity about STEM, I know that's something that I would have wanted when I was a kid. And so I'm really excited to just being able to just Zoom and try to inspire them in their biology lesson. But I'm also doing another organization for the women in science and developing world. So when I talk about my grandma being my scientific role model, oftentimes we do talk about, what would it have been like had I been born in El Salvador? And she tells me I probably would have been doing the same thing. And at first, it was really shocking because I was thinking, really, what opportunities are out there? But actually, through this program, there are so many scientific communities that are in underdeveloped countries that exist where we can connect with each other. And so right now, I'm trying to build a United States network so that we can bring scientists from Guatemala or Honduras, or we can bring them from Iraq, and we can all kind of collectively just talk together about science and what it meant to find our identities within science and how it harmonizes with our lives and aspirations. And so with that, I just want to say that in this meeting, we really are just a part of each other's journeys in life and in STEM and beyond. Um, Remembering that don't take things personal when people are embarking on their own journeys and their careers. There are often times where I don't really respond to emails or I'm really busy. But if someone were to reach out to me at any point and I knew them from class and I knew them from a past event that I did with them, I am more than happy to connect, because ultimately it takes all of us to really shape the new face of STEM. And so, again, I just want to thank you guys all for coming here and for organizing this event. And I'll be happy to answer any questions you guys may have.
Crystal, you're on mute.
I'm so sorry. Just realized that. Thank you so much, Stefanie. I just realized I don't have any questions so far in the Q&A section. But this is the time to kind of ask some questions to our presenter if you have any. Yes. Did I see a hand raised? Oh, wait, no, that was a clapping. Okay.
I mean, it can really be about anything. Honestly, like it doesn't have to necessarily be about school, but it can be about family. Like I'm a pretty open book, so.
I'll go to ask a question. It's not letting me in the Q&A because I'm co-host. But I just wanted to ask Stefanie, what does your day look like in a PhD program, and how are you able to sustain yourself? Just because I know I don't know, I'm not sure if everyone here is a little familiar with PhD programs, but sometimes you can't work, you can't have like a part-time job because a PhD, it's my understanding, right? It's kind of like a full-time job. So how does that look like in your everyday life?
Yeah, so on that note, I didn't know this, but PhDs are free. It's not like undergrad. So I get paid to go to school, which I think is awesome. And I think it really fits the kind of lifestyle that I have. And what it looks like is, one, communicating with mentors. I think it's really important to set expectations just so I know what am I doing for the next few months, especially because my PI is an MD PhD. So he goes to the clinic as a doctor, and then on other days he's here in the lab as a scientist. And so it's really interesting that he kind of lives this double life. Um, but with that being said, all mentors are different. For me, really, I'm not really compatible with a mentor that's consistently there all the time. I have a sense of accountability and intrinsic motivation. And so if I ask him for a list of things to do, I'm like, okay. And then if I'm sitting at my desk and I don't have an active experiment, I'm reading papers about how to understand my research better because I don't have an immunology background. So I'm still consistently learning. My first year was with classes. And so my second year and beyond, there's no more classes. I'm just doing about my research. And yeah, that's like pretty much it. Mhm.
Okay, so I do have some questions now in the Q&A. First question, would you recommend someone to try and go for a master's after their bachelor's to get into a PhD program, or would you tell them to try and get into more research after their BA or BS?
Yeah, that's a good question. So I'm in a community right now where a few of us were technicians after our bachelor's, a few of us went to postbacks, and a few of us went to masters, and a few of us were straight from undergrad. So I say that to say that really research the programs that you want to do. I really enjoyed my post-bac, and I also really enjoyed my industry experience. I know that that's not possible to do both at the same time. I was just put in a very unique situation that was particularly challenging, and I sacrificed a lot of my personal life for that. Um, but really trying to fit what works for you. If I think one of you mentioned before a part-time job, if you really feel like you need to save money before school, an industry experience doesn't hurt anyone, especially if you don't know if you want to stay in academia. You can pursue something in industry just to kind of get a flavor of that. But ultimately, do research on the program. I recommend probably making an Excel sheet and seeing what each program is going to do for you to shape your future aspirations if your ultimate goal is to do a PhD.
Great, thank you, Stephanie. Another question we have is, what's the thing you enjoy most what's the thing you enjoy most in lab, that you do in lab?
Oh, that's a good question. Um, I guess I guess working with human cells, that kind of sounds a little like bland, but it's true because I worked with fruit flies before. I've worked with computational skills before. Being able to work with like patient cells for whatever reason is really interesting to me. It creates this connection of like, whoa, this is a human cell. Very cool. And then I've generated my own data before and have looked at it. And so that's something that I really enjoy seeing is the whole process of how something is done. And I also like learning from my mistakes. I have definitely already made a few mistakes here and there. But I like being in an environment where people are like, oh, it's okay, you're still learning, it's totally fine.
Okay, we have another question. What is it like to be queer and in a STEM environment?
That's a very good question too. This is my advice for everyone that is figuring out their identities, maybe haven't fully come out or are still trying to discover who they are because life is it's a lifelong process of discovery. Um, I think being in a STEM space already, compounded with the fact that you're a woman in STEM, you're first generation, socioeconomically, you didn't have the best stability in that world. And then if that's coupled with being queer, oftentimes we do feel even further marginalized. But what I can say is that when you look for programs and you're interviewing or you're getting to know the school, find out if they have a queer center. I think that's really, really important. If I'm also a huge proponent of you can choose your family. I think oftentimes in our cultures, we're often preached that, our blood is our family. I don't necessarily find that to be entirely true. I have found family in people that I don't think I would have ever met if I didn't stay in STEM. Um, and also just take the time to be gracious with yourself. I think when you're looking for these schools, when you find these queer centers, and then when you are touring the schools and you really start to feel a part of everyone's energy, you'll know when you fit. And I think if you are still struggling with trying to find space in STEM, You can email me and we can definitely have more in-depth conversation about what the step been like as I was navigating this.
Great, thank you. Okay, so I have another one. Have there been any serious roadblocks, such as difficult professors or overlapping schedules to participate in opportunities?
Yes. There's a story that I openly share with everyone. When I was an undergrad at UCSC, I had this PI who I've had for 3 years, and I told her I wanted to go to graduate school. And she told me it would be embarrassing if I applied. So just kind of taking that moment to sink in. She was a woman of color too. So that was very shocking to see that she was someone I had already looked up to. Um, but in a way, I didn't need to prove anything to her because I made my peace with that. That was something that I did tell her was that, are you afraid that if I make a mistake in graduate school, that it would reflect badly on you? And she said yes. And I just remember saying, like, but it's not about you, it's about me. So having this open communication with your mentors in a way that's tactful, right? I feel like sometimes when we're telling stories to our friend, we're like, oh, but I really said it like this in my head, but I didn't say it. Like, actually take the time to really craft what you want to say, because I think having that composure and being able to deliver something really shows that you know who you are, you know who you want, and this is how you're going to do it. But also coming off as humble. I don't want you to ever go out there and say that you completely know what you want to do. I think if your PI is telling you, hey, it's probably not going to work, you can just say, yeah, you're right. Like, I mean, or maybe I haven't thought of it like that. So continuing to be open minded. So that was definitely one experience that I had. And then as far as overlapping schedules, I will say it was my industry and post-bac experience, especially because one was in the East Coast and one was something I had to drive to Silicon Valley for. And there, if my industry job was a 6, 7 hour day, I'd have to come home and spend 5, 6 hours on my post-bac. And so that became draining after a while, but I kept seeing the finish line there. Like I was gonna get into graduate school and I knew that it was going to help me. But I think time management, Google Calendar, if that doesn't help, which I just started discovering Google Calendar and I'm like, wow, this is actually great because I haven't used it. Because I used to just use a notebook and that really helps me. I carry around this tiny notebook and not the planners. I feel like I always lose planners. Like I'll go to TJ Maxx and I'll end up losing them. And so I just use like a tiny notebook and I'll just carry it with me. Like, oh yeah, this is a very important thing I have to do today. Or even just taking 10 minutes in the morning and waking up and saying, what do I have on my plate?
Great, thank you, Stephanie. I can totally relate to the TJ Maxx planner part. Okay, so I think this will be our last question, unless anyone else has another question. What is the biggest lesson you've learned as a first-generation Latina navigating higher education and or the current job market?
The biggest lesson I've learned is that I will always need a mentor because as much as I am a mentor, the way I mentor has been through the experiences that I've had in the past. I think oftentimes when we are in these marginalized environments, we try to be self-sufficient, like I mentioned at the beginning. You really try to be a self-made story. And it's not going to work. I can tell you all that it's not going to work. I think when you can reach a space within yourself to say, hey, it's okay to ask for help, that is the most incredible, kindest thing that you can do for yourself and for others, especially as you're navigating this. And remembering that your queer, Latina, intersectional identities that you may carry is what is going to make you such a phenomenal scientist, because there are going to be kids that are going to be looking up to you in the future, or people that read your science papers and they're like, oh wow, they kind of look like me, they kind of look like my aunt. And it's moments like that where you realize you don't have to separate your culture and STEM. You can be unapologetically yourself, and it's totally fine as long as you are asking for help, as long as you're being honest with yourself and who you are. I think that's going to be how you navigate the workplace.
That was great. That was great. That was a great answer. Okay, so we do have another question. This will probably be our last question for today's event, you guys. Do you have any advice for people who are more interested with field work rather than lab work?
Field work in terms of environmental biology? Yeah. So just like I have the National Institutes of Health, there is the National Atmospheric Oceanographic Association. I'm not quite sure if I'm saying that right. So it's NOAA. So it's NOAA. You can check out if they have opportunities there. And to get into field work, if you're done with your prerequisites, I would say to take a look online and look at environmental organizations out there. I'm not saying you even know, I don't even know what the environmental organizations are, but if you really just start looking and then making a list for yourself on a weekend, take that time to realize that that is another form, I think, of self-care, because you're also watching out for your own future. And so self-care can take on many forms. So do that. And then if you feel like, okay, I found an opportunity, I need to start crafting an application, I can help with as many components as I can because I don't do field work. But also maybe opening up your horizons a little bit, because even when you do field works, there are times where the specimens are actually brought back into a lab and you have to do molecular biology to analyze them. And so if you feel like you can't get your foot in the door in field research, there's just, you're getting rejections or you feel like the opportunities are out of reach, looking into opportunities in just different fields of STEM will also help, because ultimately it's underneath the realm of STEM, right? So
Great. So thank you so much, Stefanie, for taking the time out of your schedule to talk to us about your journey. I'm sure our students found your talk to be incredibly inspirational and motivational for them. I know it did for me. I also want to thank you for giving us your email to give to our students. It'll be in the chat, you guys. I posted it earlier in the chat. So feel free to reach out to Stefanie. Once again, thank you again, Stefanie, for joining us. It was great to hear from you again, and we wish you all the best in your doctoral program.
Oh, thank you so much. Yes, I'm really excited, and I hope that we can connect in the future to do an in-person visit. So great. Thank you so much again.
Awesome. Thank you. And thank you all for joining us during your lunch hour today. Thank you for your questions. Please be on the lookout for the next STEM guest speaker event on Tuesday, November 26th during Common Hour. That will be in the new Science and Engineering building. So I hope you all have a restful and relaxing weekend. Thank you again for joining us for today's event.